Thank you for coming back so peacefully. I'm very excited about this morning's speaker. Dr. Jonathan Lakey is on the cutting edge of research for type 1 diabetes. His resume goes on, and there's an awful lot of stuff that I don't understand, so I have to read it because these aren't the words I use most of the time. But he is the Director of Research at UCI's Department of Surgery and an Associate Professor at the Biomedical Engineering and head of the Stem Cell Research Center. He graduated from Alberta and received a postdoctoral training in Indianapolis. He is former director of the Comprehensive Tissue Bank. He has started worked with Dr. James Shapiro and worked and developed what's called the Edmonton Protocol, which was a huge leap in the treatment of diabetes. He's been published all over the place, both with medical journals, on magazines, on TV. He is the spokesman for type 1 diabetes, and he's been recently accepted at the position of director of the Clinical Islet Program at UCI here. So please, put your hands together and welcome Dr. Jonathan Lakey.
Thank you very much and good morning. So thank you for the opportunity to come here this morning and to speak to you about some of the research that I've been involved with over the years and some of the research that we're doing right now, and then conclude on some of the research we'd like to do in the near future. So I'm going to give you a little bit of a background on diabetes, although I think most of you probably know about the disease and the burden of diabetes. It can affect up to 6% of the population. It's the third most common disease. It's the fourth leading cause of death. The incidence in worldwide, there's approximately 130 million individuals, rising over the next 20 years to over 300 million individuals with either type 1 or type 2 diabetes. Half of these patients with type 1 diabetes will develop some sort of kidney disease, many of them leading to dialysis and kidney transplantation. It's the leading cause of blindness and amputation. The costs in the United States, and these numbers are low, about $160 billion a year are spent on diabetes and diabetes-related complications. Worldwide, it's over $350 billion annually. So that's the economic and burden of diabetes. Now, before the discovery of insulin by the Canadian team of Banting, Best, McLeod, and Collop, somebody when they were diagnosed with type 1 diabetes, they were basically placed on a starvation diet and starved to death. So the group at the University of Toronto were working on trying to find this hormone, insulin. And what they were able to show is that when you can give a patient this extract that they extracted from pancreases, diabetes became now a chronic incurable disease. And then the secondary complications, the blindness, the amputation, the premature cardiovascular disease, all became evident. So these are some notebook photographs from the University of Toronto archives. And before it was called insulin, it was called McLeod serum. It was his research laboratory. He was the boss in the University of Toronto lab, so he was able to name this work that Banting and Best were working on. And this notebook shows that some of the very early attempts at using this crude extract to inject into diabetic dogs that they had made diabetic really didn't work. It was the contributions of a University of Alberta, my old alma mater, professor by the name of James Bertram Collop. He was a chemist. He went to Toronto, and it was his contributions to use alcohol to extract the insulin from these pancreases that they were isolating to recover the insulin, that his contributions allowed this 14-year-old boy by the name of Leonard Thompson for the very first time to show that they could get the blood sugars dropping when they were given this hormone. So two interesting things from this slide. My old research laboratory in Edmonton was Collop's old lab, and Leonard Thompson's granddaughter was evaluated for an islet cell transplant in Edmonton a few years ago. So we've talked about insulin and what insulin does. So insulin lowers your blood sugar. So a diabetic that's taking insulin, conventionally, they take insulin injections twice a day, 3 times a day. I know diabetics that are injecting themselves 5 to 6 times a day. They're also measuring their blood sugar. So finger prick, measure a drop of blood, see what their blood sugars are. So this is a very labor-intensive, very costly to make sure that their blood sugars fall within a near to normal range. However, most diabetics, their blood sugars are bouncing all over. They're very lows, very highs, and it's those lows that are very dangerous. They go into the hypoglycemic comas. We also know that the swings in the blood sugar are involved in the secondary complications that I briefly talked about, the kidney disease, cardiovascular disease, and eye disease. So there are several ways to deliver insulin into the diabetic patient. There's a lot of work on developing an artificial pancreas, a system to be able to measure glucose and then secrete insulin into those patients. Those are both in closed loops and open loop systems. So there's a lot of research being done on measuring and then delivering. However, an artificial pancreas type system really is going to be limited because if you overdo the insulin, it's one of the most dangerous drugs in the world because you'd be dead very quickly. So although there's research being done in the artificial endocrine system, there's still a lot of details that need to be worked out. There's implantable insulin delivery devices. And this is an example of a MiniMed pump. MiniMed is a company, a United States-based company. It's about the size of a hockey puck, for those Canadians in the audience. It goes underneath the skin. Insulin is loaded through the septum in the middle there, and it just releases insulin over a month to 6-week period. It doesn't measure the sugar, so you have to continue to take the finger pricks to measure your blood sugar. The most physiological approach to delivering insulin into the diabetic patient is through transplantation. And transplantation can be accomplished in 2 ways. One, by transplanting the entire pancreas. Now, at University of California, Irvine, we have a pancreas transplant program. However, we only perform about 8 to 15 of those transplants every year. Why is that? One, we rely on human organ donors. And there's a chronic shortage of human organs for transplantation. Secondly, the criteria for a pancreas transplant is very strict. It's very strict because it's a very complicated procedure. Those patients are in the operating room for 6 to 8 hours. They're in the ICU for about a week. They're in hospital for at least 3 weeks. So it's a very labor-intensive, very complex procedure. A much more simpler approach is to focus on those cells within the pancreas, the islets of Langerhans. And it's these islets of Langerhans, these clusters of cells sprinkled around the pancreas that are involved with insulin secretion, the hormone insulin, the counter hormone glucagon. And it's the relationship of insulin and glucagon working together that give you precise blood sugar control. Glucagon increases your blood sugar, insulin lowers your blood sugar. However, there's other hormones within these cells scattered throughout the pancreas. The islets only make up about 5% of the total mass of the pancreas. So why transplant the whole organ when you just are interested in those individual cells? So our research laboratory, as well as many other research laboratories around the world, have been focusing on ways to isolate these cells. So we have our cadaveric organ pancreas that comes to the laboratory. We go through a whole series of steps. Usually takes between 6 to 8 hours, 3 or 4 technicians working in the laboratory to try and get the islets out of the pancreas. Here is a photograph of an isolated islet. This is called a scanning electron micrograph. Shows you and reinforces to you that we're dealing with a cluster of cells. Within each islet, there's anywhere between 1,000 to 2,000 cells. So it's these clusters of islets that we're isolating, and we're injecting them into the portal vein, which is one of the major veins that takes blood through the liver back up to the heart. And I'll show you some photographs from the operating room. And these cells are basically being lodged in the liver of the recipient, the type 1 diabetic recipient. And within the liver, these cells are able to secrete, get a good blood supply, and be able to secrete insulin within that environment. So here's the challenge. We have this. I'm glad most of you have finished your breakfast. So here is a photograph of an isolated human pancreas. This is what we start with, and this is what we end up with. For comparative purpose, a Canadian penny. So we've taken a 100 to 150 gram pancreas, focused on just those cells, and we have a teaspoon of cells that we can then safely transplant into the patient. Here is a photograph under a microscope. The islets are staining red. We have specific stains in the laboratory, so we can make the islets turn different colors. So one of which is, the islets contain a lot of zinc, so the islets stain red, whereas the rest of that tissue, which used to be the majority, is now only about 5% in that photograph. So here again is a purified human islets. This is an insulin needle, just to give you an idea of the size. The diameter across the insulin needle is about 200 microns. So again, we're dealing with a very small population of cells that can deliver and consistently deliver the insulin into the diabetic patient. So when patients come and want an islet cell transplant, we have to sit down with them. We talk about the risk-benefit of staying on insulin versus an islet transplant. We know that insulin is a treatment and not a cure. We know that there's risks of being a diabetic, the risks of kidney failure, blindness, amputation, cardiovascular disease. We know that the patients that have type 1 diabetes, their lifespan is shortened by 10 to 15 years. We also know that this subgroup of patients that we're focusing on have severe risks of hypoglycemic comas. Those are those comas that when their blood sugars get too low and they collapse. We also know that there's risks of islet cell transplantation, as in any transplant procedure. There's risks of bleeding during the infusion. There's risks of within the liver. There's also the risks here, these drugs. Because when we're transplanting islets from a cadaveric organ donor into a diabetic patient, those patients need to be placed on lifelong anti-rejection drugs. And these drugs prevent the body from destroying the islets, but they also have complications. These patients get increased risks of pneumonias, of infections, and things like that. So it's really a balancing act to know which patients should receive the islet cell transplant and which patients should continue on insulin therapy. In Edmonton, we had over 2,000 patients that were evaluated, and only about 6% of them actually went through the process and got transplants. Why only 6%? Well, there's lots of criteria of why they didn't fit the very strict criteria of this developing therapy. As we move forward, if we can find ways that we don't have to rely on human organ donors, if there's ways that we can transplant without these drugs, then we can start transplanting patients earlier on. And that's a lot of the research that I'm doing now is saying, okay, we've shown that the Edmonton protocol can work. We've shown that islet cell transplants can get patients off insulin. I'll show you some of those slides. But we need to be able to move that forward so that it can treat the millions and millions of diabetics. So here's our islet transplant process. As you remember, I said that a pancreas transplant, they're in the operating room for 6 to 8 hours. They're in hospital for weeks. What we do is we stick a small catheter or needle through the side after a little bit of anesthetic. We access this big vein, the portal vein. You can see the needle here. We run a small tube down, and here's my colleague in Edmonton, my good friend James Shapiro. We draw the islets up into a syringe. Here's me in the younger days. And we simply infuse those islets into the portal vein. The patient's awake, talking to us, and we stop insulin therapy. What happens is that here's the blood sugars of a patient before the islet cell transplant. And this represents the time of day from midnight to midnight the following day. So 24-hour period. This is their corresponding blood glucoses over a month-long period. Each dot represents the person pricking their finger, taking the blood glucose, recording it. We'd like to see their blood sugars between the blue and the red line. So this is a person that's taking insulin 3 or 4 times a day, and you can see that they have a large number of blood sugars in the upper end, but more importantly, they have some very dangerous low blood sugars. After the islet cell transplant, they continue to measure their blood sugar. They continue to record this data, and this is the type of response that we can see. So it's very encouraging to see that you're not seeing those low hypoglycemic recordings. Most of their blood sugars are falling within that time frame. And you can also see that there are some blips. It's probably after a meal. Look at, after postprandial, so after dinner, their blood sugars jump up a bit. But these islet cells that have been transplanted in these patients are functioning and have functioned for a long period of time. So we transplanted 7 patients in Edmonton, and then we were able to publish the results of those first 7 patients in the New England Journal of Medicine, and it described our technique. We described methods of how we isolate the islets. As also, we described a new potent-free immunosuppression strategy that eliminated steroids. Steroids are a common anti-rejection drug, and so we came up with a new novel protocol that allowed us to transplant without steroids. Still, we had some anti-rejection drugs, and here are their names: sirolimus, tacrolimus, and clizumab. However, we were able to see in those patients that we were able to get them insulin independent. Since that time, these are the numbers of centers worldwide that have attempted the Edmonton protocol. You can see over 60 institutions and now over 2,000 patients have received the Edmonton protocol, which we developed in Edmonton. The program in Edmonton is now over 130 patients, several centers throughout the United States. University of California, Irvine is developing a program, and we hope to have that up and running soon. In the last month, we've had the FDA come visit us, which is always a stressful time, but we came through with flying colors. We have our state approval, so we're going to begin in the next months starting our islet cell transplant program. So complications of the procedure, as I said earlier on, no surgical procedure doesn't come with complications. There's risks of bleeding when we're putting the catheter into the patients. There's risks of the cells being clogged up in some of the small veins within the liver. There's rises in liver enzymes. However, these patients are awake. We don't do this procedure in the operating room. We do it in the radiology suite. So it's a very simple procedure, and patients are going home the next day. So the indications for this transplant are patients with type 1 diabetes. We're focusing on the sickest of the diabetics, patients that have had the disease for at least 5 years. They are unresponsive in terms of C-peptide, which is a hormone that's detected in patients with type 1 diabetes. They have been placed on intensive diabetes management, and despite that, they have at least one severe hypoglycemic episode in the last year requiring third-party assistance. They have reduced awareness of hypoglycemia. They don't sense when their blood sugars are getting low, and that can be a potentially fatal complication, especially if they're driving or so forth. These are just the criteria that we've had to develop to try to make this therapy successful in these patients. So if we look at data, insulin independence data. So these are patients that have received islet transplants, and we combine for this graph data from our group in Edmonton, the group in Miami, and the group in Minnesota, the 3 most active islet cell transplant programs worldwide, and we put our data together. Again, at the time of transplant, all patients were able to achieve insulin independence. However, over time, these patients at 1 year, you still had 80% success rate. So these patients were able to be free of insulin at minimum for at least, in this graph, 4 to 5 patients were able to maintain insulin independence at 1 year post-transplant. If we look, though, over time, we see that this graph continues to show a decline, so that at 5 years, only about 1 in 5 patients are completely insulin-free. That doesn't make it not successful, because these patients continue to secrete insulin, and they have a corrected hemoglobin A1C, and they haven't had any of those secondary complications, those hypoglycemia. So why is this graph showing this progressive decrease in being insulin independent? And again, insulin independent, I'm saying that a patient that has to go back to taking insulin, maybe once a day instead of 4 times a day, we have to find a way to deliver the cells and enough of the cells so that we can keep these patients long term. And so what our lab and other labs are looking at is, why do you see this loss of function over time? Could it be related to immunosuppression? So are there issues happening in terms of the body and the drugs that we are using to fight the body from destroying those cells? In type 1 diabetics, the body, for some reason, has chosen to attack itself and attack its native islet cells. Is there something that's being reactivated? Are we going through a rejection episode? Are there non-immunological factors? So are the drugs that we're giving these patients toxic? Are these patients developing resistance to that insulin? Meaning, over time, do you have to secrete more and more insulin to see the effect? Or the islets basically being exhausted? Because when we're transplanting these patients, we're putting in as many islets as we can isolate from these cadaveric organ donors, and maybe we're not putting in enough. So they represent challenges and opportunities, and as I said, there's huge numbers of opportunities in the organ donor side, in the islet isolation side, in the engraftment and culture of the cells, as well as in drugs. So our work has been focused on the donors. We're trying to find a way so that we don't have to rely on human organ donors. We're looking at different alternatives. We're looking at stem cells. We're looking at xenotransplantation, using pig islet cells instead of human islets. We can grow pigs. They're consistent. They're reliable. Every human organ donor that we receive, it's different. There's no consistency. The way the person had died, their age, other factors. So there really was an inconsistency. What the Edmonton Protocol proved is that we have a procedure that can proof of concept. Now we have to improve it so that we can take it to the masses of people. So one of the strategies to try and avoid immunosuppression, these anti-rejection drugs, which I think played a huge part in the long-term success of the Edmonton Protocol, is to be able to put cells into a device. And here's just some examples of some encapsulation device. There's macro, large devices where you put them into the body, and then after a period of time you seed them with the cells, you put the cells, and those could be pig cells. They could be stem cells that we put into here. So the whole goal of encapsulation is to protect the cells from the body, eliminate the need for immunosuppression, and ability to retrieve the cells. So when we're working at the stem cell building, we're trying to grow cells, insulin-producing cells from either stem cells or other cells that we're working on. We have to be able to retrieve those cells if necessary. Because one of the problems of stem cells and one of the limitations of stem cell therapy is that those stem cells could potentially turn into tumors. And the last thing we want is to be transplanting tumors. So the devices that we're looking at, there are macro devices. There's also microencapsulation devices. So here's an islet cell that is surrounded by this protective covering, and that covering is made out of seaweed. So we take seaweed, we purify it, and it's the alginate, that's the chemical in the seaweed that is not recognized by the body, so it's stealth and bioinert, and we can put it into we have a process where we put that around the cells, and we can adjust chemically the size of the pores, the holes within this alginate, so that insulin and glucose can freely cross this, but some of the large molecular antibodies and things are blocked from going in. So nutrients, oxygen freely passes, but it protects the cells. So we're working with a group trying to do that. We're also working with a company where instead of putting the alginate into a capsule, we're putting it into a sheet. So about the size of a business card. We fill it with cells, and you can see the cells here. And in this example, we're putting it and adhering it to the side of the liver. There's also strategies where we try to do a conformal coating, so it's right around an alginate-type compound, directly around the islet here. So those are some of the strategies that are being explored. So again, the alginate, it comes from seaweed, chemically purified, and here we have an encapsulated piglet islet that we've developed here at the University of California, Irvine. Here's the process of encapsulation. We have a machine that's been developed. We use this alginate. What it does is it puts the cells in with the liquid alginate. It runs it through this electrostatic generator. As the droplets fall, they land into a solution of calcium chloride, and it changes it from a gel to a solid. It's really quite cool technology. So we have that encapsulated islet that we can then transplant, and we can transplant without drugs. So here's a cartoon of the islet sheet. Again, we have the alginate on either side. We have the islets in the middle, and then we can put that into. So that's almost like a tea bag, is the way I call it. And so we're now proceeding with large animal trials. We're making pigs diabetic. Surgically, we go in, we take out the pancreas, we let them heal, and then we're putting these pig islets into sheets and putting them into the diabetic pigs. So those are ongoing studies right now that are being funded by a foundation in San Francisco. Here's an example of a pig islet sheet being implanted into a diabetic rat. So we have rats that we make diabetic. We make the rat diabetic. His blood sugar goes up. We transplant, and then over time his blood sugars come down, and his body weight starts to increase nicely. We then, at 30 days post-transplant, we take out that sheet. Here you've got your islets, and you've got that explanted sheet. This is work with with the islet sheet. We're developing biocompatibility, showing that it's safe. We've done the functional studies in rodents. We're doing the large animal studies that I just briefly discussed. We're working with a group at Cedars, a metabolic lab that's going to help us validate these cells. And our goal is to put these encapsulated islets into patients in 2013. So the other source of cells are piglets. And so because of the inconsistency and challenges with human islets as a source of tissue, we've focused on xenotransplantation. And the most obvious source for xenotransplantation is the pig. Pig insulin and human insulin only differ by one amino acid. Pig is ethically the most suitable choice for xenotransplantation. And I should probably not have such a cute picture of a pig, right? In the United States alone, over 250 million pigs are used in the food industry every year. And it represents an opportunity to address the issue of chronic shortage of human organs. Pig valves are used, pig skin are used. However, there are still ethical issues of xenotransplantation. There's also issues of disease contamination. There are specific diseases and viral diseases. So you've had to develop specific disease pathogen-free herds. So we have a herd of pigs in a farm at an undisclosed location in the Midwest, where these pigs have been raised now for 4 generations. These pigs have been tested for some of these viruses, and every case to date has shown no viral transmission. So we have a herd of pigs that we're going to be using for this procedure. So we've dealt with and are addressing this whole xeno issue. There is also a company called Living Cell Technologies. It's a New Zealand-based company, trades on the Australian Stock Exchange. Its symbol is LCT, Living Cell Technologies. And they're using a similar technology to what we've developed. They're using very young neonatal pigs, isolating the cells, putting them into a capsule, a different formulated alginate capsule, and are putting it into patients. One of the unique things about Living Cell Technologies is they have a herd of pigs. Rather than having 4 or 5 generations of pigs, they found a herd of pigs in this very small island called Auckland Islands, south of New Zealand. And these pigs were left in 1863 when Captain Cook was sailing around the South Pacific. He was an explorer, not a pirate. So what these explorers did is they would leave a couple pigs on these very remote islands so that if they ever had problems, they could come back to that island and they would have an opportunity to have a food source. So these pigs have been on this island since 1863. They found them about 20 years ago. And they've been inbred, so they originally left 22 pigs. So now, here's a photograph of these pigs. They've Darwinian changed to the lifestyle. This is a very remote, uninhabited island. So when they went and started testing them, they found none of the viruses, none of the things that they're concerned about. And so they have this herd of pigs. There are no xeno-relevant viruses, parasites, or bacteria. They've removed some of these pigs to their own facility. They've had it tested by the New Zealand health authorities. Bottom line is they have a source of pigs, and these are their pig islets that they're putting into these capsules. And again, this is the scanning electron micrograph of their capsule. But believe me, here's their process. The pancreases are removed under sterile conditions, they're isolated, and they're transplanted into patients. So this is a clinical trial of encapsulated pig islets that is happening right now. So they've developed some criteria for their clinical trial. They want to see improving the blood sugar, the decrease of frequency and severity of the hypoglycemic episodes, and they'd like to have seen some insulin, reduction in insulin requirements. So from the website, I was able to get a lot of information. They did some pilot trials in New Zealand where they were able to put these capsules. Here's a laparoscopic photograph of these capsules in somebody's abdomen. 9 years later, they're able to still see these capsules inside, so these capsules survive within the body. They did a trial in Russia where they put 8 adult type 1 diabetics. They put about 10,000 islets into these patients. There were no adverse events. In terms of efficacy, they showed improved blood sugar control with reduced hemoglobin A1C, reduced number of insulin injections. 2 of their patients, they claim, were able to be completely free of insulin for up to 32 weeks, and they were able to retrieve some of the capsules after 6 months. They showed also in a second trial in Auckland where they, again, 8 patients received varying doses of islet cells. They showed that the severity of the hypoglycemic events was completely eliminated after this therapy, and that is what brought a lot of the patients. Now, in terms of insulin requirements and changes in their insulin injections, they didn't share a lot of that data. Again, being a private company, they don't have to. But for the most part, they are showing and commenting that there is reduced dosing of insulin. However, I don't think any of the patients are completely off of insulin. So our company that we're working with is called Islet Sciences. It's a United States-based company. We've done rodent studies of this encapsulated piglet technology. We've done studies in rodents. We've done primate studies in collaboration with the University of California, Davis, where we showed efficacy up to 18 months. And our goal is to have an IND, which is an investigational device approved by the FDA, to initiate a clinical trial at the University of California, Irvine in early 2013. So we've acquired key technologies for the islets and the encapsulation. Progenitor cell therapies is our GMP manufacturer. Everything that's being done has to be done in clean rooms. And so this is a company that's based in a New York-based company, but they have a laboratory in Mountain View, California, where we'll take our pig pancreases, we'll process them there, and we'll bring the cells down to Irvine for transplantation. Bottom line is we want to be in clinical trials in 2013 of this technology. So the last thing I'd like to just briefly touch on is the final source of trying to address the lack of consistency of cells, and that's an unlimited, that's stem cells. So in terms of stem cells, there's been a number of publications that have been out there, people talking about embryonic stem cells that secrete insulin, and this was a photocopy of a paper published about 12 years ago in a very prestigious journal that showed animal-made diabetic transplanted with these stem cell-derived islets, hold them for 60 days, take out the cells, the animal becomes diabetic. Sounds pretty good. However, nobody's been able to repeat that. There was also a study at the National Institutes of Health laboratory where they took embryonic stem cells, differentiated them into insulin-producing cells. They were positive for some of the markers of insulin. They looked like islets. They had a three-dimensional structure. However, when they were transplanted into mice, they couldn't correct hyperglycemia, so they didn't function. So in the end, what was discovered is that this group had cultured their stem cells in an environment with a lot of insulin, and it was basically detecting the insulin and not new growth of the cells. There is a company called Novacell down in San Diego in La Jolla that has been working on human embryonic stem cells and has been working very hard at trying to get human embryonic stem cells into insulin-producing cells. Data from 2005 showed that they were able to make these cells, however, they were relatively inefficient and had very little insulin being secreted. So the cells that they were making could produce insulin, they just had a problem delivering it and releasing it. Subsequently, they published a paper in 2008. Again, nice progression of the data showing now they have these cells, they have the transcription factors, and they've been able to show now that they can protect against hyperglycemia in diabetic animals. So it's a long road. That's the story with the stem cells. It's someday we're going to have a stem cell that secretes insulin. We're not there yet. The focus in the immediate 10 years probably will be xenotransplantation. So in summary, there's been significant progress over the last decade. There's a high rate of insulin independence using human islets and human-derived islets. High 5-year result in terms of these cells are functioning up to 5 years, but insulin independence is not maintained in all patients. Some of the patients are having to take small amounts of insulin, a third, quarter of what they were taking pre-transplant. So none of the patients that we've ever transplanted has complained about the procedure. They all see the benefit of improved blood sugar control. Good safety profile, but again, these drugs that these patients have to take are the limiting effects to taking this technology into many patients worldwide. Future opportunities, improving the way we isolate, trying to develop more potent, friendly immunosuppression, trying to eliminate immunosuppression altogether through biomaterials, trying to find an unlimited source of insulin-producing cells, either xeno or stem cells. And in terms of acknowledgments, I'd like to acknowledge the University of California, Irvine and the Department of Surgery, my clinical collaborators, Clarence Foster and David Imagawa, my research collaborators, Elliot Botvinnik, especially for a lot of the work we're working on together in terms of the biomaterials. And none of this research would be possible without funding. And our funding comes from a variety of sources, from companies. My philosophy is to work with companies because I think it'll only be a partnership between academics and companies that will allow us to get to the point of curing diabetes. Academics can't do it alone. There's just too many costs, and industry is there to support and to help a lot of this. So we are working with Islet Sciences, Islet Sheet Medical. We're also receiving grants from a number of different foundations. There's a foundation in Irvine called the North American Foundation for the Cure of Diabetes, and I've been involved with them for a number of years. We've also received money from the Hahnemann Medical Foundation, and we just received a grant from the Iacocca Medical Foundation. In the past, we've received funding from the NIH, juvenile diabetes as well. So I will welcome any questions. Thank you very much. Yes. Those are islets, by the way.
How often or how effective is a second transplant? I mean, they indicated it in New Zealand. Does that make a difference, and do you use that? And secondly, how competitive are you with the New Zealand group?
Okay, so the first question is, does a second transplant or a top-up transplant work? In Edmonton, we did a number of those procedures, and we saw a bit of an effect, but not what we would have expected. In New Zealand, they are competitive. There are some limitations of their technology. The cells that they're isolating from the neonatal piglets, they really don't start secreting insulin for about 5 to 6 weeks. The procedure that we've developed and we've patented, our islets are functional within the first couple days. So our cells will function. We don't have to wait. Additionally, our chemistry around our capsules is more robust, and we think it's a superior technology.
Hi, I'm Susie Shooter. I'm a physician, and this is fascinating work. It's amazing what you've done. I have a couple of questions. One, who pays for the patient's transplantation?
So clinically, the patient transplants have all been funded either through grants from the NIH or JDRF. So when we published the Edmonton protocol, within 3 months of publishing that results, we had a $25 million grant from the Juvenile Diabetes Foundation. So that paid for us in Edmonton to do the 100 patients that we had done.
So it's not
Subsequently, every lab has to come up with the funding. It's not a Medicare-approved procedure yet. They still consider it experimental. So for us to take our clinical protocols, I presented 2 different ones, one with the islet sheet, one with Islet Sciences. To take those protocols forward, they're going to be funded by industry.
Okay, thank you. And then also, with xenotransplants, we're using animal products. Do you still need the same immunosuppression?
So with the xeno products, they're going to be in the either the sheet or the capsule, and so our goal is to not give any immunosuppression.
And then speaking of immunosuppression, I do know that type 2 diabetes is more of a receptor cell problem, and then type 1 is really more of an autoimmune problem.
Yes.
And would it be something that anyone thought about in preventing it from progressing when it the first point of onset?
So the concept of someone with type 1 diabetes, which is an autoimmune process, the body for some reason starts to decide they're going to start attacking the beta cells and destroying them.
Is that usually with the stress? A very strong stress for a
Yes,
person,
there's stress or viral, environmental triggers, environment. There's incidents and pockets where type 1 diabetes is a much higher incidence. Okay, more time then. So there is the potential of transplanting somebody immediately upon diagnosis. However, you're putting that patient on immunosuppression as it currently stands. So we would like to get to a point where we can transplant without drugs, so the islet's in the device, and be able to have a better window to be able to treat the patient before they start to develop the secondary complications, before, there is this window effect where it's called the honeymoon, right? So there are still some residual beta cells. So if you can transplant somebody very quickly upon diagnosis, you may be able to extend the honeymoon period in a type 1 diabetic. Oh, sure. That's what they look at. Yeah. So there have been a lot of studies where a child that's newly diagnosed with type 1 diabetes, they go in immediately and put them on immunosuppression to try and preserve what little beta cells they have left. Yeah.
Hi. I have a question about the role of diet and nutrition in diabetes, and do you monitor that with your patients and how that plays? Because you haven't mentioned that.
So all of the patients, as part of their pre-screening process in Edmonton, they went through social evaluations, they met with nutritionists, they met with the endocrines, and then they met with the surgical people. So they had a group of probably, I think it was 15 different individuals that they would meet with before actually moving forward. So the nutritional side of things is very important. After the islet cell transplant and getting these patients off insulin, we were very cautious with them about protecting those cells. Don't go out to big dinners and stuff like that.
Just a quick announcement. The musicians did not show up this morning, so we have extended this time for another 10 minutes. So there's no rush. Ask this question. And if you have another topic you want to throw out, go for it.
I don't have to sing and dance, though. Yes?
Oh, wait.
Oh, sorry.
Okay.
You guys.
Yeah.
Okay.
I just wanted to know, is there ever a possibility of doing a living donor? Because when I went through
Sure.
because they said because of the extensive testing you have
Yeah.
to go through.
So a living donor would be a parent who has a child with diabetes and wants to donate a portion of their pancreas to their kid. It's been attempted once. A group in Japan took 2/3 of a pancreas from the mother and transplanted it into the daughter, who was a type 1 diabetic. The daughter had so the daughter had to be on immunosuppression because they're not identical. So the daughter had some periods of insulin independence. The mother, unfortunately, became surgically induced as a type 2 diabetic. So she as a result, got diabetes. So that was attempted once. Yes.
I think I'm the only one in here who has a pig valve. Anybody else want to step up? And I'd like to thank the hogs for saving me.
Yes. No more bacon.
And I have a microphone. I'm curious about the difference between the research in Canada and Edmonton versus here, and is there any involvement is it private funding in Canada, and is there any involvement with the Canadian medical system versus
Sure.
medicine
So
here?
in Canada, we have government funding for, like the NIH, so we would apply to those organizations. We had grants from the Juvenile Diabetes Foundation. The JDRF has branches in Canada. So there is a little bit more institutional support for research in Canada as opposed to the United States. Here you have to perform, and if you don't perform, you pass on, I guess. So, we're actively looking at a variety of different funding. We look at philanthropic organizations. We look at, some of the larger grant funding agencies like the JDRF, the NIH, the Helmsley Foundation is another one that we've just put in a grant for. We've been successful now with 2 IACOCA grants. So it's a challenge, no question. There's a lot of work that we'd love to be able to do. There's people that I'd love to be able to be involved in our work. It's just the lack of funding right now, and it's a very tough time. The NIH budget continues to decrease. We had a grant that was funded in the top 11% of the grants submitted, and it didn't get funded. The NIH funded only the top 92%, so we missed out on that one. So we're constantly writing grants as when we're not writing papers.
One more question, if I may.
Sure.
If one is not a candidate, I'm thinking of a child in Cranbrook, if one is not a candidate at this time and remains on, or if one were a candidate, see, I'm getting confused. I'm thinking of the drugs one takes instead of insulin and the dependence on those. It sounds as if, number one, there could also be a cost factor in those, and the side effects of them. Where is the balance
Well,
of
there
that?
is certainly a balance because insulin works very well in a lot of patients. However, it doesn't prevent over time the secondary complications. The anti-rejection drugs that are used and are developed by the big pharma, they have a lot of complications, and those complications come about over time. So, we'd love to be in a position to be able to transplant without drugs, and that's why a lot of our work on the engineering side of things, I think, will be successful. Yes.
I'm curious, Jonathan, about the interface between corporate work and the university. How do you work that out in terms of who owns what, and is that a complicated procedure, or is it something that's in process?
Yes, it's complicated. However, it's at UC Irvine, there is a Office of Tech Transfer. So what we've done is if a company comes to me and says we've got this technology we want to evaluate, we meet with the Tech Transfer Office. We come up with a plan, a research agreement. Any intellectual property has to be discussed ahead of time so that if we come up with a new patent, who owns the patent, who pays for the patent, all that stuff is all discussed prospectively rather than retrospectively, because retrospective becomes more of a nightmare. So we have to have very clear projects and very clear guidelines, and that's where the UCI is good in that sense and keeps me straight and narrow and keeps the companies happy too.
Now, is that true of other universities as well? I mean, is this something that's happening across the country?
I think what's happening with the lack of the big NIH grants, I think researchers are starting to see that the only way to get some of this technology evaluated is to partner with industry. And UC Irvine, over the last year, we've developed what's called Tech Portal Orange. So up at the medical center, just above our lab, there's a space called Tech Portal Orange, and it's basically been set up by the Vice Chancellor of Research to have the relationships between a UCI researcher and the business people, and for whatever project, it's got dedicated space, dedicated resources, and they handle a lot of the paperwork.
Thank you.
So maybe I missed something here. The pigs, can you take these islets, or does the pig have to be destroyed to get them?
The pigs are sacrificed, and we collect the pancreases. We're working with a couple other companies so that when the pig is sacrificed and we take the pancreas out, we're working with a company that takes the liver, and they're going to isolate the cells from the liver. They're called hepatocytes, and they put those for another project. There's people that have talked about getting the pig hearts for pig valves. We're trying to make best use of that tissue from those pigs. So we're hoping that we'll take pig skin and use that, because that's used in a lot of burns. So people that have had a burn, they use, and then thyroid as well. So, you know, we try to make, you know, no bacon, unfortunately. No. Okay.
And is there going to be you know, if this really starts big, will there be enough supply of these pigs that don't have all the diseases?
Yeah, so it takes about 18 months from the time you decide to start a pig facility till you actually have pigs available for transplantation. So the company that we're working with, Islet Sciences, we will, as soon as we start getting our approvals through the FDA, we have a big lead time. And so, you know, I anticipate there being 20 or 30 of these pig facilities around the United States in close association with the academic institutions that will be doing the transplant. So it's going to be a big ramp up, and costly.
I just want to say, this is such a complex thing, from pigs to donors to what works, and you know, one word, fantastic. Thank you so much.
Juggling a lot of things. Thank you.
That's it. Thank you so much. Thank you. This is fantastic.
Irvine, and so that's part of my mandate, is to educate and to